So it turns out when you transplant young bone marrow into old mice, the old mice live about 30% longer and they're younger in every way that we check. Many other labs have shown this result as well. And so, I've been on the investment side for many years, close to a decade, and I thought I'd seen it all. I know what are the largest effect size interventions, which ones are well reproduced and legit. And this is the largest effect size uh, and safest approach because it's your own HSCs, it's autologous. Um, so this is what caused me to, of the many things I could have been doing, to focus on rejuvenating the bone marrow. The other reason it's really safe is because, as I said, it's autologous, meaning it's your own cells. And so the way our long-term vision here is the immune system reboot where basically you go into any clinic, outpatient procedure, you get an injection of a molecule that's widely used for bone marrow transplants or donations today that causes the HSCs to circulate in your bloodstream. And then you apherese them out, you pluck them out with a simple machine, and then you have isolated your HSCs just through a blood draw, effectively. So you don't need to go into the hip bone or anything like that, as happened some years ago. So this is an outpatient procedure. We isolate the HSCs, we expand them. We have a unique expansion method that our collaborators in Oxford, University of Oxford, published in Nature twice that allows a thousand-fold expansion of HSCs, maintaining their stemness, their stem-like qualities. So we can isolate them, expand them, bank them for later because no one's getting any younger. You're going to want those in the future. Then we rejuvenate them with our proprietary cocktails and reinfuse them periodically, almost like an oil change or a tune-up in your car. We refresh the immune system. We gently sort of replace the immune system over time. And the analogy to this is the ship of Theseus. So the ancient Greek myth about the concept of continuity. You replace some pieces of the ship over time. When do you decide that you have a new ship? So that's our goal, the immune system reboot. And I think that this is the lowest hanging fruit, most likely way to extend human lifespan in the near term.
Hello, Sebastian. So you are the founder and CEO of ImmuneAge Bio, as well as the co-founder and general partner at Healthspan Capital. So welcome to Modern Healthspan, and thank you so much for joining us today.
Thanks for having me. Good to be here.
So Sebastian, so you're very active actually in the investment community and have founded a number of companies. So it was difficult to actually create a an intro with only two of them. But those are the two kind of important ones and the ones that I kind of want to talk about today. So if we, I mean, and I certainly do want to talk about like the market and healthspan capital and where the money's going. But if we could start with ImmuneAge Bio, because I guess that one is kind of closest to your heart because you're actually the CEO of it. Could you give what is the goal of the company?
Sure. So at ImmuneAge Bio, we're the first drug discovery platform for immune system rejuvenation. So we target hematopoietic stem cells, which are found in the bone marrow. These stem cells give rise to the entire immune system. All the various immune cells that surveil for tumors and pathogens and orchestrate tissue regeneration, the whole immune system derives initially from the bone marrow in the hematopoietic stem cell, the HSC. In addition, we're also testing small molecules that rejuvenate other immune cell types, including T cells, which are a very hot cell type in the oncology space as well as the autoimmune space. So, what we do with our collaborator lab in Switzerland, in Lausanne, Switzerland, we have a pretty unique drug screening platform where we can look at many different hallmarks of aging in HSCs and T cells, potentially other cells in the future. So we can do relatively high throughput screening in these cells, looking at the hallmarks of aging and function. And we can do it in an unbiased fashion. So we can basically take various omics inputs and classify using deep learning, relatively straightforward AI method to tell us, which cells are truly younger. And then we do bone marrow transplants and other assays to assess their function. So yeah, thus far we've been focusing a lot on mitochondrial function. So HSCs are the fastest dividing cell type in the whole body. They produce hundreds of billions of cells per day. And so red blood cells as well as white blood cells, immune cells. And so they're very energy hungry, kind of like the brain. The brain is 5% of the body mass, 20% of the energy burned. The immune system is similarly hungry for energy. And so therefore, if anything goes wrong with the mitochondria, you notice it in the immune system relatively quickly. So it turns out when you transplant young bone marrow into old mice, the old mice live about 30% longer and they're younger in every way that we check. Many other labs have shown this result as well. And so, I've been on the investment side for many years, close to a decade, and I thought I'd seen it all. I know what are the largest effect size interventions, which ones are well reproduced and legit. And this is the largest effect size uh, and safest approach because it's your own HSCs, it's autologous. Um, so this is what caused me to, of the many things I could have been doing, to focus on rejuvenating the bone marrow. The other reason it's really safe is because, as I said, it's autologous, meaning it's your own cells. And so the way our long-term vision here is the immune system reboot where basically you go into any clinic, outpatient procedure, you get an injection of a molecule that's widely used for bone marrow transplants or donations today that causes the HSCs to circulate in your bloodstream. And then you apherese them out, you pluck them out with a simple machine, and then you have isolated your HSCs just through a blood draw, effectively. So you don't need to go into the hip bone or anything like that, as happened some years ago. So this is an outpatient procedure. We isolate the HSCs, we expand them. We have a unique expansion method that our collaborators in Oxford, University of Oxford, published in Nature twice. It allows a thousand-fold expansion of HSCs, maintaining their stemness, their stem-like qualities. So we can isolate them, expand them, bank them for later because no one's getting any younger. You're going to want those in the future. Then we rejuvenate them with our proprietary cocktails and reinfuse them periodically almost like an oil change or a tune-up in your car, we refresh the immune system. We gently sort of replace the immune system over time. And the analogy to this is the ship of Theseus. So the ancient Greek myth about the concept of continuity. You replace some pieces of the ship over time. When do you decide that you have a new ship? So that's our goal, the immune system reboot. And I think that this is the lowest hanging fruit, most likely way to extend human lifespan in the near term.
So, yeah, a couple of questions in there. Would you, so you're going to rejuvenate the immune system. Do you think you need to also rejuvenate the thymus, or does that kind of happen by itself because you need to teach the T cells?
Yeah, so thymic rejuvenation would definitely be complementary, and there are a bunch of groups working on this. I think it's perhaps in some ways an easier problem. But the cool thing about HSC rejuvenation is that there are progenitors that go from the HSCs to the thymus and rejuvenate the thymus. So if you actually transplant young to old bone marrow, there's improved thymic output in the animals receiving the young bone marrow. So yeah, the health of the thymus depends to some extent on the health of the bone marrow. But yeah, I mean, starting in our 20s or so, we get thymic involution shrinking, and I have a friend at Oxford who works on this like mathematical modeling of thymus function, and he pointed out that could be pure coincidence, but he pointed out that the incidence of cancer increase correlates pretty much perfectly with thymic output, thymic function, which makes sense, right? You have T cells constantly surveilling for tumors. So yeah, rejuvenating the thymus, I'm a big fan. There are a number of groups working on that right now.
Your rejuvenation of the immune system, is this with small molecules, or how are you doing that? Because I saw that one of the products you're working on is like a pill to rejuvenate the immune system, which seems like such a no-brainer that people would want because the immune system is so central to so many things. Um, where are you on that pill? And is that the same thing that you use when you do the autologous stem cells?
Sure, yeah. So we have an in vivo program or an orally available small molecule program as well as an ex vivo rejuvenation program. Um, so there's a single agent oral small molecule that enhances mitophagy, mitochondrial biogenesis, There's a lot of data on structurally similar natural products. We did medicinal chemistry, made about 300 analogs of a certain natural product, and made more drug-like versions of this compound. So that would be the pill. And obvious, there are many indications that you could pursue for that, right? You could have chronic infectious disease, you just have normal aging, and you could give it before cold and flu season. You could give this pill before receiving a vaccine to improve vaccine response, T cell and B cell function in elderly. And then, this molecule enhances mitochondrial function across the board. So there are neurodegeneration applications. There are applications in basically every indication. So it's truly a pipeline in a pill. Also, the natural product version of this compound extends lifespan. A group at the Buck Institute showed it extends lifespan, a little over 10%, and that's not even with optimal dosing. So it's a bona fide geroprotective drug. And then that molecule we combine in a cocktail ex vivo with about four other agents. And that we believe will be a lot more powerful because those other agents address other hallmarks of aging. So, as we've seen in medicine, generally it's better to have a cocktail approach than a single molecule, right? Think about chemotherapy. Generally, it's not a single molecule today. It's, four different compounds or so. And so, or the cure for HIV or semi-cure for HIV is three or four compounds. Or, sorry, the cure for hepatitis C rather is multiple compounds. The treatment for HIV, which is pretty good, is also multiple compounds. So we've seen that for the most efficacious therapies, the ones that could actually be curative, we need more than one small molecule generally. So what we're doing is basically culturing the HSCs and T cells in a cocktail of several molecules. We can't do a hundred different molecules because it just becomes logistically complicated from a regulatory perspective. You could do that if the regulators weren't standing in the way and could have better effects. But it's just you have to keep it simple for them. You also have to keep it simple for GMP manufacturing. So currently we're focused on small molecules, easy to screen, easy to manufacture, but there's no reason in principle for the ex vivo program, at least. We couldn't do all kinds of molecules, peptides, proteins, RNA, DNA, whatever you want. Even gene editing. If you could do multiplex CRISPR engineering on the HSCs, that's actually going to be a longer lasting effect than the small molecules, right? I mean, we can incubate HSCs or T cells in small molecules. It gives them kind of a boost. It resets things to some extent. But once that molecule washes out, the effects aren't going to persist forever, right? So unless it flips some kind of transcriptional switch, which some of these molecules are. But if you just engineered the cells, and we know certain mutations that are really beneficial to engineer, that will last the life of the cell, the life of the HSC once transplanted. And that can be, in HSCs, that can be your whole life. In some HSC clones, they last lifelong. So to give you one example of the power of CRISPR engineering the HSCs or the T cells, this has been tried by a couple groups in the past, sort of before CRISPR came along, but basically engineering these cells to be resistant to HIV. So it's happened that patients with HIV have been cured from a bone marrow transplant. This has been published in Nature a couple times. It's rare, but it occurs where the recipient of a bone marrow transplant gets bone marrow from a lucky person who happens to be resistant to HIV. Something like 1 or 2% of the European population has this mutation. We know what some of these mutations are. One is CCR5 that the Japanese professor CRISPR'd into the embryo some years ago. But there are a handful of these sort of super controller mutations. And so, in principle, we could isolate the HSCs of an HIV patient. We could CRISPR in two or three of these mutations that make them resistant and then put them back. And those HSCs, in principle, would outcompete the other HSCs that are still vulnerable to the HIV or the helper T cells that are produced. And so that will cause viral load to go down over time, provided that the HSCs you transplant are outcompeting the native unengineered HSCs, which seems likely. And there are other ways to guarantee that that happens. So, so we could effectively cure HIV in that way. Um, and there was a group probably 10, 15 years ago, a biotech company that attempted this with T cell engineering, but they did it with zinc fingers and it was just 15 years ago, technically complex. The other thing is very expensive. We'd have to get the cost down in order to do HSC engineering for every patient, but we're doing that now for sickle cell anemia, right? I mean, this has been approved in the UK and the US. It's called Casgevy. It's a HSC engineered sickle cell product, and they're charging, I don't know, something like $1 million for it. But that is actually a good deal for the healthcare system because these sickle cell patients over a lifetime, not only do they suffer immensely, it's a painful disease, but they're expensive to the healthcare system. So it actually ends up being worth it even at that price. And that price will come down over time. I mean, it doesn't need to be that price. That's mostly amortizing the cost of the failed R&D budget and the risk to reward investors. But over time, once that, therapy goes a little more mainstream, the price will come down, as is the case for pretty much every drug.
Something I've been really digging into recently is how we can protect our DNA as we get older. And there has been some fascinating research coming out of Australia about magnesium. They found that when we're low on magnesium, it leaves our DNA more vulnerable to damage. Meanwhile, up to 80% of us are walking around with low magnesium levels. To maintain optimal levels, I supplement with magnesium, and I specifically choose Magnesium Breakthrough by BiOptimizers. What sets it apart is its comprehensive formula, containing all 7 highly bioavailable forms. And while the magnesium is protecting my DNA, I've also noticed I'm sleeping better, feeling less stressed, getting better recovery after workouts, and have more energy. If you want to try out Magnesium Breakthrough, they have a 365-day money-back guarantee. And by visiting bioptimizers.com/modern and using the code MODERN10, you will get a 10% discount. If you subscribe, not only will you get amazing discounts and free gifts, you will make sure your monthly supply is guaranteed. So when you're doing you're putting in the HSCs, you put them into the bone marrow and they kind of graft. Is that they engraft and then they start multiplying? Is that how it
We
works?
actually infuse them into the bloodstream and they home to the bone marrow. This is how bone marrow
Oh,
transplants work today. Yeah, we don't have to like inject them into the bone or anything like that. HSCs are actually circulating. It's relatively rare, but they're actually circulating throughout the body. HSCs and bone marrow derived MSCs, mesenchymal stem cells, as well, that have tissue regenerative properties. So it's quite hands off. We mobilize into the bloodstream from the bone marrow, apheresis machine to take them out. We expand, rejuvenate, bank, then reinfuse periodically into the bloodstream, and they home to the bone marrow. So it's quite non-invasive, low invasiveness. This is something that can be scaled massively to the general population. I can imagine, once it's proven to work, you can imagine it being part of your employment, healthcare perks package, right? Periodic immune system rejuvenation in 20 years. One of the near-term applications, though, is oncology. So one of our investors and advisory board members is Thomas Ebeling. He's the former CEO of Novartis. Novartis is one of the largest pharmas in the world, one of the more sophisticated, based in Switzerland, and they're one of the leaders in oncology. And when he was CEO of Novartis, he saw firsthand that chemotherapy, traditional chemo, does a lot of damage to the patient, right? Like if the chemo doesn't, you're trying to kill the cancer before the chemo kills the patient, effectively. So chemo, it does DNA damage, mitochondrial damage, and so it leaves you in an aged state. It reduces immune function because HSCs are a really fast dividing cell type, the fastest dividing cell in the whole body, in fact. And chemo selectively kills and damages fast dividing cells that are replicating their DNA quickly. And so the dose limiting toxicity on chemotherapy that prevents you from going with a higher, more efficacious dose is bone marrow toxicity, myelosuppression. So doctors have to monitor the bone marrow function before in titrating the dose for cancer patients. So in principle, what we could do here, and this has actually been done in some small clinical studies previously, is bank the HSCs before you get the chemo. They dodge the bullet of the chemo, and then we reinfuse them post chemo, preventing tumor relapse, which is due in part to the damaged immune system post chemo. The chemo damages your immune system. That's why the tumors come back in the future. The same tumor and new types of tumors, and a lot of the other long-term side effects of chemo. And also, hopefully, improve in the short term the recovery of these patients, preventing infection in the hospital and things like this. So this has actually been done in a small study in Stanford. They didn't rejuvenate, they didn't expand, but there were pretty significantly positive outcomes. And I spoke to a bunch of hematologists about this. Hey, do you think this would work? They say, yes, we believe that this will work, but we don't do this as a standard of care because we don't get reimbursed by either the government or insurance for providing that procedure. And if it goes wrong, we get in trouble. So there's no incentive for us to do this because no one has run a large enough clinical study to definitively prove that it works such that we get reimbursement or such that it gets on the government approved list. So this is one way I saw firsthand, and I see firsthand as an investor all the time, in which the government, perhaps well-meaning, is suffocating innovation because throughout most of history, it was doctors who were experimenting. They had a little bit more leeway to try things with patients' consent, and discover new medical advances. Throughout history, a lot of medical advances were just doctors trying new things, drugs in new diseases or in new dosing regimens or whatever. But, in the spirit of safetyism, we're constraining the ability of doctors to actually discover new medicines, for better or worse.
So one of the things that I see is if you're going to have a drug an intervention, such as the one that you're making, which would rejuvenate the immune system, it needs to be priced at a reasonable level because people are going to take it prophylactically over a period of time. And they're not facing imminent death or like some disease. So, but the whole system, the way the FDA works and the approval process kind of forces the intervention to be very expensive. Do you see that? Do you agree with me? And do you see any way around that?
Sure, yeah. So there are significant regulatory costs that drive up the price of drugs because we have to run very large, very long studies, and we have to pay some millions of dollars to comply with regulations. It's kind of an arcane Byzantine system, and not just in one country, but in multiple countries. Generally, what is done is companies get approval in the US, FDA approval. That's seen as quite rigorous gold standard, and then all the other countries sort of follow from FDA, but not always. Sometimes the EMA doesn't, and they diverge. But yeah, like I know I have friends who have gotten drugs to market, and they pretty much invariably say, we could have done this 10 times cheaper if we didn't have to comply with all of the specific requirements of regulators. We could have done smaller, faster studies, doing them in other countries, for example. And that would save a lot of money for the healthcare system because you don't have to recoup as high costs. But that said, with our approach and with many geroprotective drugs, especially the orally available pills, where it's just simple. You pop a pill, you don't have to do anything ex vivo with cells. That can be really cheap because the indication size, the number of patients taking the product, allows you to price it more cheaply because the market is big enough to recoup your risk and R&D costs. Whereas if you have like a very rare disease, in order to justify the costs, you have to charge a lot. So that's why a lot of these gene therapies and cell therapies cost hundreds of thousands or millions because it's in a rare disease population. It's not just that gene and cell therapy manufacturing is so expensive. It is currently pretty expensive, but it's not that expensive. So it's just to do with the small patient population to justify the costs. But that's one of the great things about aging, longevity. It's kind of bad and that everyone's aging, but everybody will want the product ultimately. So you don't have to price it as expensively.
Now that does have some hope. Do you think there's any? So one of the problems, one of the expenses I see with the system with HSCs is it has to be autologous. So each person you have to take something out, play with it, put it back in again. Is there any chance of making like non-allogenic HSCs that you could make young and then create in a bioreactor?
Um, yes, in principle, I mean the technology is not really there yet, but in principle, you could take skin cells or blood cells and turn those into iPSCs or embryonic like stem cells, and then convert those into HSCs. Nobody has achieved that yet because iPSCs, induced pluripotent stem cells, Yamanaka reprogramming, that's there's a lot of hype, there's a lot of excitement, which I think on a long term is justified, but in the short term, it's pretty far away. The iPSCs, for example, the conversion rate is very low, so it's not an efficient process. And then when they do convert, they have a bunch of weird mutations like P53 mutation, loss of function, which is the most important gene preventing cancer, right? So you don't want to transplant in these sort of precancerous type cells that are very stem-like and have the ability to grow a lot. So eventually, I think we could make them from iPSCs, but it's not a big deal to do a peripheral mobilization of your own HSCs. That's not a big deal. That's done in 90 something percent of all bone marrow donations. So I've signed up for these um, organizations, nonprofits that try to match you with a donor. And when you do that, you ask the donor to come and donate their bone marrow. Years ago, you'd actually take a needle and go into the hip. It's not a big deal. It's under local anesthesia. It takes 15 minutes, but you feel some pressure. It's like not pleasant. But with this peripheral mobilization, it's pretty routinely done all over the world every single day. So that's not really that big of a problem, I would say.
And then they can use your process to multiply these and rejuvenate them, would be the
Yeah, the
plan.
more HSCs you have in a transplant, the more effective that transplant will be. So it's the number one factor, along with patient donor age, that determines bone marrow transplant success. So many bone marrow transplants are done with, a certain level of HSCs available, and if they can't get that level mobilized from the donor, then they don't proceed with the bone marrow transplant. Uh, but we find that, this is demonstrated in the literature, the higher you go in terms of the number of HSCs that you can transplant, the better, almost monotonically, linearly, are the bone marrow transplant outcomes. And it makes sense because that new bone marrow has to engraft in your bone marrow niche. So to some extent, the more the better. And a lot of transplants just don't proceed if you can't get enough HSCs. That's with an autologous, or sorry, an allogeneic transplant. So from somebody else,
Mm.
another person's bone marrow, and that's complicated. Um, Autologous transplants are done in some indications, but a lot more rarely. But they're quite safe because they're your own immune system. They're not going to react against your tissues as someone else's immune system, even if they're a well-matched donor, might. And so for allogeneic transplants, which are mostly done for example, leukemias or severe autoimmune diseases, you have to be on immune suppression for some period of time. And so there's a risk of graft versus host disease. It's not a trivial thing to do an allogeneic bone marrow transplant, but autologous is extremely safe by comparison.
So this pill that would help you rejuvenate your immune system, um, kind of where is that in the process of making it to the clinic? Could you talk about
Sure.
that?
Yeah, sure. So we've done a bunch of medicinal chemistry on it. We're probably about 2 years away from the clinic currently. We need to do some more in vivo testing, with some of the early molecules look great in vivo in our hands and a bunch of other labs' hands. These new compounds, we want to run through a handful of other models, infectious disease, cancer model. We've done bone marrow transplant. And then we need to do IND enabling studies, which generally take about a year, and then we should be ready for the clinic at that point. And the lead indications currently are preventing cold and flu during cold and flu season, which is actually a major cause of death and disability. There's nothing available for it. So before cold and flu season comes around, older patients will take the pill and prevent hospitalizations. Another indication is vaccine response, and we can get conditional approval pretty quickly for that, where maybe someone's getting the flu vaccine or shingles or some other elderly patient getting the vaccine, we give the pill maybe starting a month before and then monitor T and B cell function. You can actually check how many immune cells have responded to the vaccine, and you can have that readout within a couple months. So that's a pretty quick, cheap study to do.
Yes, and and those kind of indications, like I don't want to get a cold. It seems like you would be able to persuade your doctor to prescribe that to you if you wanted.
Yeah, for sure. And a lot of people will probably use it off label because
Yeah.
it has a lot of other benefits. Like people report taking similar molecules that work similarly, improved exercise endurance. A lot of people I know are taking a natural product that works kind of similarly. We've modified this molecule. So, and there are a handful of clinical trials with that natural product that show, reduced inflammation, improved insulin sensitivity, a whole lot of other effects in many different indications. So, the odds are high that people will use it off label for other indications as well.
Yes, I think I would use it. It sounds like really cool.
Me too.
Okay, so your other job you is as part of Healthspan Capital, right? Your general partner, and heard you talk about something called LongBio. So, what is LongBio?
Yeah, thank you for asking. This is a term I'm trying to popularize with the team at LongBio Fellowship, Longevity Biotech Fellowship, which is the largest organization in the world for scientists, founders, investors, and anyone keen on longevity in a very serious way. It was co-founded by my colleague Nathan Cheng, who is one of my partners at Healthspan Capital. So the reason we're trying to promote this term is kind of like what the term synbio did for synthetic biology as a field is like a very distinct, concrete subsector of biotech that VCs need to have a thesis on, that VCs need to be informed about, and that they can specialize in, as well as just sort of concretizing it as something that's truly distinct from just biotech broadly. Um, it also just saves time. It's faster than saying longevity biotech every time. So, so Healthspan Capital, we are an angel fund. We're the most active investor in longevity biotech and regenerative medicine. To our knowledge, we've done about 32 deals in the last 2 to 3 years. And we invest generally quite early. We try to help out the founders, being pretty hands-on, helping with hiring, connections, raising capital, etc. Before that, I was a principal at Apollo Health Ventures. Apollo is today the largest aging-focused fund. It's more of a company builder model based in Germany. It's about 300 million under management and one of the more sophisticated investors and builders in the field. And we co-invest with Apollo. And after that, I was at Cambrian Biopharma. I was the co-founder and chief investment officer of Cambrian Biopharma. This is one of the larger multi-asset long bio companies based in New York City. We have two assets in the clinic, an mTOR inhibitor out of Novartis that is selective for mTOR complex 1 over complex 2, so mitigating the side effects of rapamycin, led by Joan Manick, who was at Restor Bio and Novartis previously. And then we also have amplifier therapeutics, which is an AMP kinase activating small molecule. And AMP kinase is kind of one of the holy grail targets in the metabolic obesity, cardiometabolic space. Pharma has tried to drug it a couple times, didn't succeed, but the team at Amplifier out of Sweden has actually managed to do it, and Cambrian has partnered with this company to get it in the clinic, run some studies in diabetes and obesity. And this molecule, AMP kinase activation, actually causes fat loss without muscle loss in the animals. So this is very important compared to, say, the GLP-1 class of drugs and many other obesity drugs that are kind of effectively caloric restriction mimetics in the sense they make you eat less. But when you do that, you tend to lose muscle as well. Whereas the AMP kinase activation, it almost just makes metabolism look like a young person, right? Like we all know young people, teenagers who are just eating constantly and they don't get fat. Whereas with age, we tend to store fat more readily. And that's actually, I think, a feature, not a bug, evolutionarily, because as you age, as a hunter-gatherer, you're not going to be out there hunting and gathering as much. So you're going to want to store more. whereas the younger people, they consume that energy in doing things. So there's actually some metabolic switch that occurs, many people in their early 30s, where they just realize they have to count calories, etc. And so that switch back, this is part of the evolutionary mismatch or theory that explains so much about modernity and how we're sick and have other problems, evolutionarily mismatched to the environment in which we evolved. Hunter-gatherer society, lots of physical activity and community and all kinds of stuff. And so part of that evolutionary mismatch is that we're still eating a huge amount of calories as older people that we wouldn't have done as a hunter-gatherer. The most of the calories would have gone to probably the younger people hunting and gathering. Anyway, so flipping that switch back, adjusting to the evolutionary mismatch. And I actually think a lot of drugs are doing exactly that. They're kind of compensating for evolutionary mismatch. They're either they're either like all the weight loss drugs, right? It's or sleeping pills, for example. We're exposed the evolutionary mismatch. We're exposed to all this bright light at night, all of this activity and all of this thinking and job anxieties or whatever, causing us to sleep poorly. So we have to take these sleeping pills in order to compensate for that. So it's another case of pharmaceuticals as sort of lifestyle medicine mediating that evolutionary mismatch. But then the other class of drugs, in addition to infectious disease drugs, are just aging drugs. That's not necessarily an evolutionary mismatch, it's just a decay process. So I think most drugs can fit into some of those categories. Um, anyway, so back to LongBio. Our thesis is that LongBio is going to be the most important sector of healthcare eventually. We just don't know exactly when, right? Everyone's aging. Aging drives 90% of disease burden. Probably don't need to preach to the choir of your audience about that. And yet, almost no capital and attention is going to it. There are a lot of newspaper articles talking about longevity, mostly the sort of diet and lifestyle type stuff, Brian Johnson type stuff, which is great. Like people should care about their health, but the sort of unfortunate realization I've made is that even with the perfect, most ascetic diet and lifestyle, and assuming we get it correct, there's a lot of debate about what the perfect diet and lifestyle are. We still would probably only get maybe 10% lifespan extension in humans. Because extrapolating from mice, if we put them on the most extreme intervention, which is caloric restriction of like 30%, maybe you get 30% lifespan extension. But the mice are not very happy. They're hangry, they're cold. So 30% lifespan extension is too much. 10 or 20% lifespan extension, maybe you get 10 or 20% extension in the mice. But generally, what we find is that longevity interventions that work in simpler organisms have a smaller effect size in more complex organisms and organisms that are already longer lived. And humans, relative to mice, are evolved more optimized for longevity to begin with. So it will be harder to move the needle. If you look at, um, you know, other primates, primates maybe they live 30 years or something, humans live 80s. And we can look at the genome differences between us and other primates, and we actually know the pathways and the genes that are conferring longer life. And they've already been optimized to some extent, like autophagy, inflammatory response, DNA repair fidelity, etc. The hallmarks of aging have actually been mutated, have been optimized in humans versus other primates. And so the question is, has the low-hanging fruit been picked in the human genome? And is it going to be harder to eke out more longevity gains because of that? Um, and and I think that's true to some extent, but I think there's a loophole. There's a caveat. And this is, I think, original to me, but probably someone else has had this idea previously, as far as I'm aware, which is the loophole is we harness evolutionary mismatch to our favor rather than to our detriment, as it's been. What does that mean? So calories have always been scarce and rate limiting in human evolutionary history in all animals, but now they're not. So what if we could engineer the genome to basically not store fat, store calories as fat, which cause a lot of problems, and leverage those calories for repair processes. So instead of transforming that energy from calories from food into reproduction and growth, which kind of persists throughout aging. Like your cartilage keeps growing, your internal organs keep growing, and it kind of causes dysfunction. And causes cancer, that growth impulse. But away from growth and reproduction toward repair. And this is the disposable soma theory, basically, and coupled with the antagonistic pleiotropy theory of aging. So if we could just somehow engineer the genome or take drugs that would reroute energy, caloric input, into repair rather than storage or growth and reproduction, then we would have more resources to invest in, say, DNA repair or autophagy, proteostasis, all of the other processes that are keeping us well. Um, so anyway,
Yes, because I mean that is the calorie restriction, and like turning on autophagy is one of the things that's like well one of the things that rapamycin does that seems to work. And
yes.
yeah, I mean if you could just flick that switch more easily without having to go through all of the kind of
Yeah, the body
trauma.
resists autophagy. The body resists undergoing autophagy. I know autophagy pretty well. It's one of my favorite areas because I co-founded a company called Samsara Therapeutics in Oxford, England, in collaboration with Apollo Ventures, which is, as far as we're aware, the most advanced autophagy enhancement small molecule drug discovery platform. So we have several compounds that activate autophagy in an mTOR independent way, different from rapamycin, and and safer than rapamycin. And so that it would be great if we had pills that would induce autophagy because the body resists it. It's really hard to do extended fasting for most people. Evolutionarily, the body really doesn't want to do it, and cells resist turning on autophagy because it consumes resources. It's an energy intensive process. So you actually have to input energy to break down the cell and then rebuild it. It's pretty energy intensive. So the whole machinery of life, and that's why I think it'll be hard to engineer, but worth trying, is geared toward conservation of energy, conservation of calories, because they're scarce in the environment, but no longer. So if we could just coax the body into using the hamburger that we just ate to repair the genome rather than store it as fat, that would be great. And I take inspiration from Bruce Ames. Bruce Ames invented the Ames test, which is standard in all toxicology to assess the mutagenicity of a new compound. He is a professor at UC Berkeley. He is the National Medal of Science winner. He's like 100 years old. He's still doing science. One of the greatest living biochemists, Bruce Ames. And he has focused a lot on nutrition, nutritional biochemistry. And he came out with a paper some years ago on the triage hypothesis. And the triage hypothesis was in PNAS. It was a review. And the basic idea is micronutrients like vitamins and minerals are sequestered and selectively used to their most urgent application rather than the application that extends lifespan most. So, for example, let's say you consume some magnesium. The urgent application is to use it in muscle contraction for your heart beating or something like that, or maybe for function of the immune system. Only when you have a supraphysiological amount or an orthomolecular dose, as Linus Pauling, the two-time Nobel laureate and organic chemist, called it, you take a super high dose of these vitamins, will they be used for the urgent use, but then they have a surplus, they have extra, they can be reallocated to repair and long-termism. So you have this short-term, long-term trade-off with nutrients. And because the nutrients are scarce in nature, they tend to go to the short term first. And once that has been resolved, only then will they be used for DNA repair or long-term preservation of the soma. So, what do you do with that information? Okay, you take high doses of vitamins and minerals and stuff like that, but that's only going to get you so far. So the next step would be, can we find ways of engineering ourselves or using drugs to basically tune the use of these nutrients, whether they're macronutrients like proteins, carbs, fats, calories, or they're micronutrients like vitamins and minerals, how do we direct those resources toward long-termism and repair of the soma rather than the short-term applications and storage of calories? So nobody's
Yep.
really working on this right now. I mean, it's still kind of science fiction, but I think that is a way that we can crack that problem of are we already pretty well optimized for longevity as a species within, Homo sapiens within the primate family? We're already by far the longest lived primate. And so we've selected ourselves for longevity. So how do we go beyond what nature has already done?
So thinking about where we are in terms of because you surveil the market and you're aware of most of the companies. In fact, I think you have your friend Nathan Cheng. You know, he watches the market for these and he keeps a list of all the companies. What do you see as the most the first solution that will extend lifespan beyond lifestyle? And like, how far away do you think that is?
Mm, like therapeutics that extend
Yeah, a
lifespan?
therapeutic.
Sure. Well, there are already some that I'm pretty confident will extend lifespan in most people. Rapamycin is a good example. That extends lifespan across pretty much all species. It does have some side effects, so it may not be justified for most, but there's probably a dose at which it is justified. People talk a lot about metformin. Metformin seems relatively safe, although there are some pterogenicity problems. Um, but it seems to be most of the benefit of metformin is for people who are metabolically unwell, like diabetes or not exercising. The gains seem to be lost in people who are relatively metabolically well to begin with. And then there's a U-shaped curve in the mice where if you overdose the metformin, they start to live shorter. So you have to get the perfectly right dose. And if you do get the perfect dose in mice, you get about a 5% lifespan extension based on the ITP NIA testing program. So the effect size is limited. Um, but there are a bunch of supplements out there that probably extend lifespan that have been shown to extend lifespan in various species too. But what we think is going to extend lifespan the most are gene and cell therapies. So we really like non-viral vector technologies. We like stem cell therapies, mesenchymal cell therapies, etc. So those are the areas we focus on the most, gene and cell therapy going forward.
How do you do non-viral gene therapy?
Sure. So throughout the last couple of decades, most gene therapies have been viruses like AAVs. AAVs have a lot of liabilities. They're expensive to manufacture, they're immunogenic, they can't be redosed because the immune system recognizes them, they can damage the liver, etc. So, what if we had vectors that would deliver nucleic acids? Oh, and then the biggest problem with AAVs is they're not tissue tropic, so they mostly hit the liver. You have to inject them locally if you want to hit a certain tissue. And still, they mostly end up in the liver. So you don't get a very high dose of the gene and the tissue of interest. So, so a lot of groups, one we've invested in, Ocean Bio, OISIN, is a non-viral vector technology. So you can make them out of all kinds of stuff. You can make them out of silicon, you can make them out of lipids. You can make them out of lipids and then attach a bunch of proteins to the outside to make them do other stuff. So there are a lot of these sort of nanoparticle-based vector technologies, but and they're cheaper to manufacture. They're not as immunogenic. They have a lot of advantages over AAVs, but they all still suffer from that delivery problem, getting it to enough cells in the body in the target organ of interest. That's a very difficult engineering challenge. So a number of groups are working on that, and one solution is cell type specific expression of your construct. So one way you can target tissues is to have the vector just only transduce into those tissues. The other way is you transduce into all tissues or many different tissues, but the gene of interest is only expressed in the tissue you want. And that is basically gene expression 101. There are genes in the liver that are only expressed in the liver and they're not expressed in the eyeball, right? And epigenetics is governing that process. So we can actually do that artificially, where we deliver the construct, the DNA, to a bunch of different tissues, maybe even the whole body. But say we only want to express it in the vasculature. So we find promoters, genetic sequences that naturally occur in the cell that turn on certain genes only in certain cell types, like the lining of the vascular wall, for example. And so we can actually selectively express those constructs in certain cell types. You can even go a step further and add another construct to that nucleic acid that you're delivering that is an on or off switch. So we can actually create a construct which is routinely used, kind of like TET on or off switches is one example, where you can take a pill of a certain molecule that turns on or off the gene. And so you could regulate the expression relatively tightly. There are all kinds of interesting things you can do with genetic engineering and synthetic biology type methods that are extremely powerful and precision controlled compared to the current and historical paradigm of pharmacology, whether it's a plant that you grind up and isolate some small molecules, that's what most of pharmacology has been throughout history, or you derive it from small molecule from a microbe. You pop the pill, you hope that it circulates to the right tissues, doesn't cause damage to your liver or your kidneys, but gets into your brain, which is a very difficult problem because the brain's trying to keep everything out. It's a blood-brain barrier, right? So that's small molecule drug discovery. That's the bread and butter of pharmacology, um, and the pharmaceutical industry. It's great. There are advantages to it. We know how to do it pretty well, but there are a lot of disadvantages. You can't control specific cell types with uh, small molecules, right? Or even antibodies or peptides. It's the same pharmacokinetic, pharmacodynamic process. It's kind of um, probabilistic and not very well controlled. Whereas genetics are very complex feedback systems. So you could imagine having a genetic construct. You could engineer a cell like a B cell, for example, which one of our uh, portfolio founders, Matt Schultz, who's also CEO of Ocean Bio, has done, um, where it's called ImmuSoft. This company is backed by Peter Thiel and others, and they're in the clinic now for engineering a B cell, a circulating immune cell that normally creates antibodies constantly, pumping out a huge amount of protein as antibodies, can be engineered to pump out any protein of interest. So they could produce GLP-1s, the weight loss drugs, if you wanted, or they could produce these enzymes for enzyme replacement therapy, which are used for rare genetic diseases like Tay-Sachs disease to break down these pathologic molecules that accumulate in the brain. That's what patients do today. They inject themselves with protein enzymes. It's very expensive and it doesn't work very well. So you can actually become your own protein production factory or your own nucleic acid production factory in situ within the body. Another company we invested in called Rejuvenating Bio is doing something similar. They basically came up with a lipid nanoparticle and a certain type of plasmid DNA construct you inject into your adipose cells, and then those adipose cells, just in your belly or whatever, produce whatever protein you're interested in, and you can control its expression with on and off switches like these small molecules. So, we, as an industry, we spend huge resources on these huge facilities, antibody manufacturing, protein manufacturing facilities, these huge vats take up a whole room that have yeast in them or microbes or human cells pumping out drugs, antibodies, for example. And it's very expensive. And we have to ship them places. We have to do quality control on them. It would be much better to just take a gene therapy and produce in situ in your body, your own bioreactor, producing whatever you need. So that's coming probably within the next decade or two.
That does sound wonderful. It sounds a great way of doing it. But yeah, the time frame of a decade or two sounds um,
That's the problem. I mean,
Yeah.
there's so I put a fair amount of blame on regulators, um, but it's not all their fault that this proceeds slowly because diseases only progress at a certain pace. It's kind of an irreducible pace, right? Like if you want to do an Alzheimer's study, you need to monitor these patients for at least a couple years to notice a delta, depending on the effect size of your drug, to notice a delta between those curves. And so there's just an irreducible pace that especially age-related degenerative diseases take. And aging itself proceeds, fortunately for us, at a pretty slow pace in general. Like even the same person 2 years later, if we were to measure with all of the best biomarkers we possibly could characterizing their aging, as long as they didn't get some specific serious disease in the interim period, it would be hard for us to notice that delta in any of our metrics because humans just age at a relatively slow pace, whereas a mouse is, dead or practically dead in that time period. So it's it's good and bad in that we have to run these longer studies. However, if you have a large enough patient population, you can actually get good enough statistics in a shorter and shorter period of time. So in principle, if our measurement tools like these aging biomarkers, epigenetic clocks, composites of all kinds of anything you want to measure about somebody, if that were more accurate and we could tell, on a several month basis or a yearly basis, then what you could do is you could just have a couple thousand patients in your trial, and then you could assess the pace of aging relatively quickly or the pace of decline of any disease. And this was actually the trial design of the TAME study with metformin, which is a cool study design. And FDA gave it the green light to proceed. It was like 3,000 patient 2 year study. I don't think metformin was the best shot we could take on goal. I think we'd be better off with low dose rapamycin or something like that. Um, or maybe the SGLT2 inhibitors that make you eliminate extra glucose that also extend lifespan more than metformin. Um, but the cool thing about that TAME study is that FDA approved the primary endpoint, which is latency from first to second morbidity. So when you get sick with one disease, you're likely to get sick with another shortly thereafter. Really fun chatting with you, and sorry to jump, but happy to join you again in the near future.
Okay. Thank you so much for joining us.
Thanks, Richard.
Cheers.
Have a good
Thank
one.
you. Yeah. Bye.